stress you have and how that, in a point function, how that could translate to deformation with many deformations across an entire area. So we can take that, we know what the shear stress is, we can integrate that, we know what the force is from a cell. We can put the directionality to that, get axial force, transverse force. And if I look at how much force these cells are producing, I see something interesting. So these cells are contracting to really short, and they're not producing much force. These cells are staying longer, and they're producing more force. In the heart, you have this, well, even muscle cells, but very well studied in the heart, this relationship between sarcomere length or muscle cell length and force. Where if the heart is stretched more or contracts, keeping at a higher sarcomere length, then the heart will contract with more force. And this actually is what keeps blood from pooling in the body or in the lungs, because if more blood enters one ventricle, it'll expand that ventricle will contract with more force until you end up in a steady state. Sometimes that steady state is the heart continuing to fill with blood, which leads to congestive heart failure, but generally that steady state is a place where you end up with the same output from the right side of the heart as the left side of the heart. So you expect this to keep going up. But after a week, we get less force on very stiff materials, which says that the cells are different. Look at why that less force is. It's not because there's less contractile protein. It's not because of the biophysical measurements. There's a velocity effect also, as well as a length effect, and we looked at that, and that doesn't correlate with this lower force. But if we look at calcium, calcium correlates with this lower force. Interestingly, it also correlates on this side too. But you end up with less calcium. Less calcium means you have less interactions of actin and myosin, and the heart can contract with less force. We looked at why you have less calcium, and it's because these cells don't have, in very stiff environments, don't have a well-developed sarcoplasmic reticulum. And this is an example of a Western blot Sarca, which is sarcoendoplasmic reticular calcium ATPase. It's a calcium pump. In these calcium stores inside heart cells that store calcium and release them during every heartbeat. These calcium stores develop as the heart cell matures. They aren't in very immature cells. They're always present in very mature cells. And they seem to only develop on ideal Young's modulus and not develop as you get softer or stiffer. So that was really interesting, that something's preventing this from forming. Remember I showed also previously that when we take these cells and look at, just look at them, they have these stress fibers that you don't see physiologically on really stiff surfaces. Had the hypothesis maybe the development of those stress fibers is preventing the internal cytoskeleton reorganization required to develop this sarcoplasmic reticulum, these internal calcium stores. So the great thing about stress fibers, the pathway that leads to the formation of stress fibers with an applied outside force on a cell is known. And it goes through a Rho GTPase and a Rho kinase, or ROCK. And there are ROCK inhibitors. So C3 toxin is a Rho GTPase inhibitor. This Fasidil, also has some long number name to it, is a ROCK inhibitor. And if we inhibit the cell from being able to form stress fibers, and then do the same experiment, we're looking at force over elastic modulus. And you kind of see where this is going by all the room that I leave up there. All we have to do is inhibit stress fiber formation and can recover the expected, you get to a stiffer environment, the cell deforms it less, and the cell starts producing more force. And that really gets back to in vivo what is seen with heart cells and what you would expect with a heart cell, as opposed to these, either you call it pathologic condition or just an artifact of culturing them in the wrong mechanical environments where you don't see this maturation. So now we've taken this further. I decided, because I think this group's much more interested in scaffold composition. We've done a lot of electrophysiology on this. We actually just submitted a paper this morning before I went to my first meeting today. I approved it on the electrophysiology of patch clamping these cells and looking at calcium channels and calcium activation, especially the inactivation of calcium channels. So if you have questions on that, I can answer that. But wanted to make sure I had time to get to the biomaterials part of this, scaffold composition. Right, so we want to take something that we can implant, that maybe we can put cells on outside the body and implant. Ideally, for translating into the clinic, we get something that we implant and all the cells we need come into it. I think that's possible for small corrections. I think you quickly get to a size where that doesn't become possible. But looking at these and getting to the question of, all right, so we need a patch. First place this is going to be used, no matter how great it is in routine, not routine, but current therapies. We're using a piece of plastic, and that piece of plastic has a 25% chance of calcifying or causing some sort of bend in the heart as the heart grows and the plastic doesn't grow, or causing an arrhythmia, and surgeons need to go back in to either cut it out, put something else in, or go back in to restructure another vessel coming off near it, or go back in to implant a pacemaker or automatic defibrillator, right, another surgery. And we think that, an initial success is get something that can be sutured in that doesn't become a scar. Ideally, if it's small enough and it doesn't contain cells, or if it's large enough and you put some amniotic fluid cells into it, and it just becomes part of the heart. And we need something now that then can be suturable, that can repair a hole, that can withstand ventricular pressures. So these, 200 millimeters of mercury, that can also then contain and support cells. You have a porous structure throughout, and we know if we want immature cells coming into them, we don't want a hard piece of plastic, because those immature cells aren't going to mature. And really what's going to do is recruit fibroblasts, and it's going to become scar tissue, and that's what typically happens. So we looked at different materials, started this with a multi-layered approach. So you need something that's strong tensily, but is compressible, because the cells that go into it, we want to be able to compress it. It's compressible, has a compression that's similar to native heart tissue. So we used self-assembled polycaprolactone. This figured out how to make polycaprolactone in a way where we actually crystallize it on water in a liquid in a water environment, and we can get this highly porous material that is not as hydrophobic as polycaprolactone usually is, I should say, in that we can get physical entanglement with these chitosan collagen emulsions. So we take chitin, which is the shells of shrimp. You deacetylate it. You take off an acetyl group by putting it in a high environment, a high pH environment, a very basic environment. And you could get this material then that will gel if it comes down to the right pH and the right temperature. In fact, you can keep it cold and it doesn't gel, and heat it up and it immediately gels, which makes it great for handling. Cells don't attach to it. Mammalian cells don't attach well to it. So we want to take it and put it in something that attaches cells. We start off with just an emulsion, just made in a sonicator, by emulsifying collagen with this chitosan, and found that we can make these multi-layered patches. They stay together. They don't delaminate. In fact, you actually tear apart the collagen before you tear the collagen off of the polycaprolactone. And we get compressive modulus by varying the amount of gelatin, where we can tune the compressive modulus to about that 10 kilopascal number, between 10 and 15 kilopascals, about that number that matches the compressive modulus of native heart tissue, while still maintaining an ultimate tensile stress where these will withstand ventricular pressures and a suture strength. And what's great about this way that we were making, what's really the polycaprolactone that's putting in the suture strength, we could suture it to a pig heart and then pull on it, and the suture actually pulls out of the heart muscle from the pig heart. We just get pig hearts from a slaughterhouse. Or, actually, one cool thing being in Houston, there's a restaurant right next to the medical school campus that serves lamb hearts, and it's a way cheaper, and you don't need any animal approval because you're just buying a food product. The restaurant will just sell us a whole lamb heart.
Wow.
And I'm sure, yeah, they make more from us than somebody ordering it as a meal, and we pay less to them than we would pay to any other place that would supply us fish. A master's student a few years ago figured that one out. That was really great. So you look at a cross-sectional view, we get these very highly porous scaffolds. The porosity comes from lyophilizing it with a very specific control of temperature. And if we do that, we hydrolyze it, put your neonatal rat cardiomyocytes on. We get very good, here's the surface, we get very good survival of neonatal rat cardiomyocytes. Over the course of 4 days, they migrate through the whole thing. We start to get some cell death at about 1.5 millimeters into the batch. So that kind of gives us a limit for if we don't have any vascularization, what will cells survive just from diffusion from the outside. So we've taken these, shown that we get good force, but not as good as heart tissue. Remember we're looking at how do we not have the problem of causing arrhythmia. We get good conduction velocity, but not as good as heart tissue. And the conduction velocity, in fact, is important. Yes?
Just to understand your upper graph there,
Yes.
that tree structure at the top, is that showing something about significance level?
That is showing significance, yes.
Okay,
That's
so the first two on the left are the same, is that how to interpret that, and then the next three are the same?
This is significantly different from these three, and this is significantly different from these two.
Okay.
That's what that's showing.
Okay.
Yeah.
Cool. Thanks.
Yeah, I know sometimes they end up not. You show these and don't have the caption. Yeah, so we want conduction velocity, it's just how fast, part of the heart beats, how fast that translates to the rest of the heart. If you slow that down enough, you can actually always cause an ectopic beat, or always cause an extra heartbeat. Now, if I put something in that slows down the heart, I can induce ventricular fibrillation if I put it in the right area and slow it down right, because the beat can go through and get to the other part right after it repolarizes, and you can end up with a circular waveform that follows. So that's a really bad thing to do. So we don't want to slow down the heart. So we looked at what might be able to speed it up. One material we looked at is we're just putting in gelatin, just collagen into this as a cell attachment. That's not native cell attachment. We looked at making native cell attachment, and then we started collaborating with this group at UCSD that's looking at taking heart matrix. They decellularize hearts from pigs, left ventricle, and actually going into clinical trials now, injecting that matrix after an infarct in humans. And so we got some of that heart matrix from pigs. Thought cells are probably going to like attaching to that better. And they do, you get very high viability on heart matrix versus gelatin. You actually get higher force, but you get a big increase in conduction velocity. So here, we stimulate here on a round patch, and this is just color-coded to how long it takes the signal to get across that patch, 12 millimeter patch. So here it takes 300 milliseconds, here it takes 200 milliseconds. So we're getting closer to native, but not quite to native. Thought, how can we get this going really fast? Started collaborating with a lab at Rice that specializes in single-wall carbon nanotubes. And a lot of this is inspired. There's this great paper out of Stanford that used artificial membranes that showed that if you put a carbon nanotube in the membrane, you can transmit across. You can transmit an electrical signal across that membrane. And we were looking with this lab that wanted to do some studies of these single-walled carbon nanotubes that they're looking at. How poisonous are they? Yeah, the other studies, carbon nanotubes are toxic to heart cells. Are these toxic? We looked at them. They're toxic at high levels. They're not toxic at low levels, but they fit right into the cell membrane. They autofluoresce. You can look at exactly where they go. And we thought, can these be used to really help lower the threshold for cells to fire and to really speed up a conduction velocity? So we made these same chitosan gels. This was really cool. How these were being dispersed before was with a surfactant that allows these to be dissolved and dispersed, these single-walled carbon nanotubes, because they generally just want to clump together. Right? They're very hydrophobic. It's all carbon. You're throwing them into basically salt water. They want to clump together, and the detergents that we used are all toxic to cells. The detergents this lab was using. And by this brilliant postdoc who I have, realized, he said, we're doing all this stuff in chitosan. Chitosan's amphiphilic. Chitosan has the hydrophobic region and hydrophilic region. And can chitosan be used instead of the detergent? And it can. And we could actually disperse this in chitosan. So we can disperse it. We see viability as long as we keep the concentration below about 100 parts per million. We don't get death of cardiac cells. And we put this in, we see higher beating frequency when we grow cells with these single-walled carbon nanotubes in the scaffold that the cells are growing in. And the great thing is conduction velocity. And you saw those other ones. Here, we greatly increase the conduction velocity. And we actually get to, here, conduction velocities, even at 33 parts per million concentration, that match the conduction velocity in the native heart. At the time we published this, we were the fastest conduction velocity of anybody to publish in cardiac tissue engineering. Nina Grosstock's lab at Duke has since published one that's a little bit faster than ours. But still, we're in the range of native tissue. So this is really cool. So now we're going right now into tests of these in an in vivo model. The in vivo model we're using is a rat heart. So if we're using cells, we're using athymic rats. Otherwise, we're using Sprague Dolly rats. And the reason for that is that a mouse heart is about this big and a rat heart is about this big. So you can do this surgery on a rat. We can't do it on a mouse, although the cardiac surgeon thinks we can. So we replace, what we end up doing is replace about half of the right ventricular free wall with a patch by kind of pinching it down, cutting it out while the heart's beating, putting in a patch, releasing it, and so all of this tissue bounces back, and what you end up having is this patch that's contacting blood on the inside. It's just replacing that wall. You can do this in a rat because a rat really doesn't need their right ventricles. You can just put a piece of plastic in. Also, all of our controls, we use bovine pericardium, fixed bovine pericardium that we bought from St. Jude Medical that is an approved cardiac patch used in surgeries. So we do this, put this in a rat, go for 0, 4 weeks, 8 weeks. We've now taken this out to 16 weeks. When you put just pericardium in, you get this big scar area. And when you put one with gelatin, a heart matrix in, you get an area that has some blood vessels in it and is a smaller scar area. We use MRI and look at cardiac function. Actually don't see a difference between cardiac function. As soon as you put in the patch, you not surprisingly have lower right ventricular function because you're replacing part of the right ventricle, and that comes back up by 8 weeks with any patch you put in to standard ventricular function. Yes?
How does the rat heart size compare with the newborn? Close or
Smaller. Yeah. Way smaller and importantly, way thinner. You actually don't need, so the issue with the blood vessel supply, that becomes much more important in a human. Like it's much easier to repair a rat heart. So this is where we start from, but you need to go to a large animal model.
Okay.
So we do histology on this, show through, in these multi-layered patches, the parts that were gel get muscle cells that grow through them. We get blood vessels inside the parts that were gel, and the parts that were polycaprolactone turn into get this very thin foreign body response. So this is a foreign body response, a thin area of tissue around it. We get viable blood vessels through when we use a multi-layered patch. If we just use available commercial bovine pericardium, we do not get any blood vessels that grow through. Use some more histology stains, look at fibrosis. The bovine pericardium just becomes scar tissue. The other parts have this, this foreign body response around the polycaprolactone, and the rest becomes muscular tissue, and the muscular cells grow in to this hydrogel. And so we can show this, and we can show kind of fibrous scar tissue that you initially get a big scar, and then with these multi-layered patch, the muscle tissue grows in, and the scar starts to go away. So now we've got these patches, we can put them in. The next goal, that's going on right now, is vascularizing them. What happens if they're vascularized? What happens if they have muscle cells in them that come from the induced pluripotent stem cells? And what happens if we put the carbon nanotubes into them and look at those types of effects? So the next is just going through that with a rat model, but then going to a larger animal model. Probably a sheep model, because a sheep model grows in the way that humans grow. A pig model can be about the same size, but development isn't as fast. So in summary, so I've shown this population of stem cells isolated from amniotic fluid. We can make those into cardiomyocytes going through an IPS stage. We can make them into vascular cells. We can develop these composite grafts that we can plant in a rat model and replace part of the right ventricle of a rat, and these become somewhat muscularized. And so future projects, still running short on time, but future projects that I want to do is to continue the 3D heart tissue, look at these IPS cell generation from amniotic fluid cells for repair of birth defects. Continue use of carbon nanotubes really to increase the, decrease the chance of arrhythmia in heart patches. I think other fetal interventions with amniotic fluid stem cells. And then the great thing here is both large animal models for validating this, but also the chance of working in regenerative therapy for veterinary medicine, which there's very few people around the country looking at, and there are people here at CSU looking at that. So engineered 3D heart tissue. This, I have an NIH R01 that just started at the beginning of this year on bringing in a graduate student and bringing in a couple postdocs to work on this, making 3D tissue constructs with the ability to either vascularize or put in cardiac cells and using that in a mouse model. Also looking at how do we make this cardiac tissue without having to deal with the cells outside of it and implant them. Can we just put the cells in and everything happens inside the tissue in a way that you could actually make in at a company or make in a hospital? And so looking at that, I actually have an NSF grant that started this past summer using a nanoparticle drug release So instead of us just adding factors, it gets added with either time delay or controlled photo releasable nanoparticle delay from inside materials. I want to expand, so we've studied these IPS cells, been able to make them, and are now looking at IPS cells from known cardiac defects made from fetal amniotic fluid stem cells from fetuses with known cardiac defects. And I think this really gives us the chance. There are a couple places that are looking at some specific ion channel defects, but I think that there are a lot more that may be more complex, but may lead to structural issues, where you can really look at what are the changes, what are the mutations in the cells, or what are the genes that are affected, and can you modify those and do studies on actual models of defects. Continuing with the carbon nanotube work that I mentioned, and also continuing for other fetal interventions with these amniotic fluid stem cells, particularly looking at congenital diaphragmatic hernia, where the diaphragm doesn't form, and because intestines and liver are going up into the chest cavity, you end up with poor lung formation. We think that amniotic fluid stem cells made into lung epithelial cells could help with this. And spina bifida repair, which is now being repaired with fetal interventions, several places around the US. And there still seems to be damage to the spinal cord. We think that stem cells that can be made into neuroprogenitor cells could help with that. And then I mentioned, one of the few places here which actually is looking at repair of heart defects in dogs and horses. And Chris Horton, who I've met here, is one of the very few people looking at how you can repair heart defects in dogs. Funding, so here's where I've gotten funding from in the past: NSF, NIH, and the American Heart Association mostly, with some other smaller grants. Future funding, which is focused primarily on National Institute of Health and NSF, with things maybe from Coulter Foundation once you get into a translational material. And they wanted me to mention teaching. I think I teach a lot of basic courses. My background is in chemical engineering, so I think I can teach some of the thermal sciences, thermodynamics, materials courses here in the Department of Mechanical Engineering. All of the same of engineering economy, which I think translates to most engineering programs, control systems, and then especially when you get into biomedical nanomaterials, I think that's an area. Do you not think that translates?
She was taking a picture because that's what students do in class.
I take a picture of the teaching slide.
I think I could develop a graduate level engineering course on stem cells and engineered materials and therapies, which there doesn't seem to be anything like that here, and especially with the new focus on regenerative medicine, with the new Regenerative Medicine Center. I would expect there to be much more research, more graduate students looking at regenerative medicine. So having something that ties regenerative medicine to engineering would be a very good course to have. I do have training. I've developed problem-based learning modules for every class I've taught. I have training in various design and flipped classroom, and really trying to interact with class students. I tend to get really good teaching evaluations. So that's been good. I have some evaluation comments, and I can send you this, but there are people who really like classes I've taught, but I can answer anything on teaching strategies. Graduate students, I've now graduated 3 graduate students. I have 2 in my lab. One of them's on this slide because she just joined 2 weeks ago. But my graduate students have gone on, 1 into academia, 2 into industry, and have won, the ones who've been in the lab for a while, have won big awards, NSF fellowships, NIH fellowships, many best poster competitions. I actually forgot to put Young won a best poster competition this summer, and somehow I forgot to put that on here. So many competitions like that. Postdocs, have a postdoc who has gone on, he's now an associate professor of tissue engineering at South Carolina Medical university. My first postdoc actually got tenure before I did. Have another postdoc who is, in fact, he's now officially a research scientist who's one of the best people I've ever worked with. I think would be great running his own lab. I think that's what he should do someday. But I'd like to have him come with me wherever I go. So that's it. Sorry, I think I've got exactly my hours. These are all the people who actually did all the work. So I have to thank them. Any questions? Yes.
So with your chitosan gels, when you're making those, how are you controlling geometry or anything, or do you just make a vat of it or a mold of it and cut out what you need? I know there's a lot of 3D printing.
Yeah, so everything I've shown from making them have been made in a circle. But we are controlling geometry. We're actually 3D printing molds that we can then control electrospun scaffolds instead of these PCL scaffolds that I was showing here, and then put a gel around that mold and control for geometry. And that's something that we're developing and trying to get to where we can look at effects of geometry. But that's not all that tough to do to control geometry.
Sure.
So I think that really if you're looking at for specific applications, possibly even specific patients, and if you want to build an entire ventricle, you probably are looking patient-specific at controlling geometry. If you're going to put a right ventricular outflow tract patch, you're probably not. You're not going to 3D print that. You're just going to make them all kind of the same.
Cool.
Yes?
I had two materials questions.
Okay.
Has someone analyzed the heart matrix, or like what is it made out of? They take real heart matrix,
Yeah.
but they must know what it's made out of, full
Yeah.
consistency.
So it's lots of collagen 1, collagen 4, and laminin with some elastin through it. There's a really interesting paper by Lauren Blackwood at Tufts University that came out a couple of years ago that took heart matrix from fetal hearts, neonatal hearts, and adult hearts, and actually compared their big differences compared to in this rat model on how the extracellular matrix changes through them. It's actually much more elastic in a fetal heart that tends to go away in an adult heart.
And then the collagen that you were using in your materials, it's gelatin. I mean, it's not
Yeah.
really, whereas the collagen in the heart matrix is probably more native.
Yeah.
The reason I asked is that I always think of collagen as being pro-thrombotic and pro-scarring, or encouraging that, and so I wondered why you would want to put it in your material, but maybe I'm not thinking about it.
Yeah, I think collagen's the most common protein anywhere, so I don't think you could say it's always
Right.
pro-thrombotic and pro-scarring. I think collagen tends to be secreted